So good afternoon, everyone, and thanks very much for your kind invitation to speak today. This is an exciting opportunity for me as a pediatric epileptologist. In terms of my our research, basically we're focused on understanding how epileptic activity can spread through large-scale networks, how this could result in cognitive comorbidities, and ultimately what we could do to treat or even prevent these sorts of epileptic processes. And so what I'd like to do today is share with you some of our work where we've been mapping large-scale networks in focal epilepsy, starting in animal models but extending to humans, and then our ability or the start of our work to develop responsive neuromodulation techniques to try and really interact with these networks and try and revert them towards their physiologic state. And even though I'm not using specific models of LGS, I think a lot of the principles that we're working under are directly applicable, and I'm excited to move in this direction. Okay, so I don't have any disclosures. So to start off with a little bit about the physiology of large-scale networks. So we know that large-scale networks interact via coordinated oscillatory activity, and this is thought to allow coupling between brain regions that facilitates neuronal communication, and this mediates diverse brain processes. And so you can see here what's happening is that action potentials from different brain regions are arriving in this precise temporal epoch, such that they can especially communicate, establish information flow, and promote long-term plasticity. And so this was highlighted by a very nice study where in an animal model where they looked at long-term memory, which requires a dialogue between the hippocampus and the cortex, so a large-scale network. And what they found is that if you artificially increase the coupling between two memory-related oscillations across these two structures, you could actually improve memory in rodents. And so despite the importance of these sorts of mechanisms that are increasingly being determined to regulate key functions such as memory, very little is known about how they're disrupted in pathologic conditions like epilepsy. And so, of course, as we've already very nicely heard today, multimodal studies have shown us that the structure, the function, the performance of large-scale networks, they're all affected by epilepsy. Even if the epilepsy initially seems to originate in a small focus or multiple small foci within the brain. And yet it's really unclear how these foci result in large-scale network dysfunction. And of course, clinical experience tells us that epilepsy is dynamic. Some patients actually experience remission of their epilepsy, whereas others have refractory seizures or progressive cognitive or intellectual dysfunction, such as specifically the case with LGS. And so unfortunately, we know even less about the mechanisms that regulate this sort of plasticity over time. So the specific question that we wanted to address is, how does large-scale pathology arise and progress in epilepsy? And hopefully, by doing this, we would be able to identify some biomarkers for patients at risk for progression. And of course, the most important goal somewhere down the line is to identify the potential therapeutic and preventive approaches. So I'm going to start off with a little bit of introduction to intrarectal epileptiform discharges, which is what the majority of my work focuses on. And so these are the hallmarks of the epileptic brain. They're brief, they're spontaneous, pathologic bursts of activity. They occur in between seizures, and they interrupt ongoing network activity. And so here's an example of an individual IED waveform that we record neurophysiologically. You can see it's high amplitude, and it's actually generated by bursts of synchronized firing among population of neurons. And so unlike the physiologic oscillatory activity that governs memory, for instance, these IEDs don't contain interpretable information. In patients with LGS, such as the example on the EEG that I'm showing here, IEDs are typically frequent, they're repetitive, and they can even be nearly continuous at times. And this provides even more potential disruption to the network. So we know that IEDs define the regions broadly that are part of the epileptic network, and they're also known to contribute to cognitive dysfunction in patients with epilepsy. And although the rate of IEDs can fluctuate, they're often persistent in these patients and they're frequent. And so what we hypothesized was that IEDs themselves could play a role in shaping neural network interactions that govern epilepsy progression and also potentially cognitive dysfunction. So the way that we first tried to investigate this was to look at an animal model that would allow us to induce a progressive epileptic process in a controlled manner. And this is a Kindling model. And so Kindling is an old, well-established model for primarily human temporal lobe epilepsy. But importantly, it has many good features for a model of a progressive epilepsy in general. It induces chronic hyperexcitability of neurons. It induces similar seizure types to what we would see in patients. It's associated with memory impairment and also exhibits similar responsiveness to anticonvulsant medication. And so the way that we kindle rats is we deliver brief localized trains of electrical stimulation, such as the one that I'm showing you here, and then we generate a seizure pattern. And this is an example from one of our rats. And importantly, this actually generates seizures that have progressively increasing severity over time. And in their interictal state, they have spontaneous IEDs that closely resemble human IEDs. And this allows us to focus on the progression of interictal abnormalities in this type of a model. Importantly, with our approach, we use in vivo electrophysiology, and this allows us to investigate and map large-scale networks. And so the network that I'm going to speak most about today is the hippocampal cortical network, but these same techniques can be applied to multiple different networks. So here, as an example, I'm going to speak to the interaction between the hippocampus and the medial prefrontal cortex, or the MPFC. And that's because, as you can see here, the hippocampus communicates directly to the medial prefrontal cortex, and it also has indirect connections through the thalamus, and there are many recurrent and associative feedback loops that connect the structures. And importantly, we know that the hippocampus and the medial prefrontal cortex communicate for physiologic purposes for long-term memory, and the mechanisms by which they do this have been sorted out. So this gives us a good starting place to understand how pathologic activity could disrupt these communications in a large-scale network. So a little bit about what we know already for the physiology. We know that in the hippocampus, there are these high-frequency oscillations called ripples that order neural firing and encode sequences of information. And similarly, in the cortex, there are these lower frequency oscillations called spindles that also sequence information and neural firing. And what happens is during sleep, the cortex is fluctuating between these activated up states and these dysfacilitated down states. And there are specific epochs of temporal communication where the ripples are coupled with the spindles. And the theory is that this oscillatory coupling allows information to be transferred from the hippocampus to the cortex, which is important for long-term memory. So what happens when you introduce IEDs into a large-scale network like this? So in our rats, we performed in vivo electrophysiology, as I mentioned, and we induced seizures in our rats, and then these rats developed spontaneous hippocampal IEDs, such as this one here that I'm showing you. And what was very interesting and surprising to us is that actually, after an IED in the hippocampus, you very often saw a spindle in the cortex. And if you plot just an occurrence rate of these two oscillations, you can see that they happen very frequently concurrently. And so we wanted to quantify this relationship, and we did that using a cross-correlation measure. And I'll explain this just briefly because it's important for some of the other quantification that we do later on in our human studies. And so basically what we do here is we take the occurrence time of either a ripple or an IED, and that's on the x-axis, and we look at the probability of seeing a spindle oscillation. That's on the y. And so what you can see here, this is the physiologic state. Time 0 is when a ripple happens. There's a small significant peak of increased probability that you have a spindle afterwards. And what's very interesting is when you turn to the epileptic state, if you take an IED at time 0, now you have this huge peak of very strong temporal coupling with spindles. And so this suggested to us that what we were actually inducing a much stronger pathologic form of coupling when IEDs are introduced into this large-scale network. But what does this really mean in terms of the function of the network? So to try to get at this, we used memory testing in our rodents. And so we use a task that's hippocampus and cortex dependent, and it's called the cheese board maze, and it just allows rodents to forage on a maze with hidden water wells, and they learn the locations of 3 rewards. And so what we found is that kindled rats were able to learn the locations of the rewards just as well as normal rats. So here we plot how the rat moves across our maze for the last 5 trials of the training session. And this is for a normal rat, and you can see it comes out of its home box, it finds the rewards in a stereotyped manner very efficiently. It knows exactly where the rewards are. Similarly, for a kindled rat, knows exactly where those rewards are. In contrast, if you come back after 24 hours, you put the normal rat on the maze, it remembers precisely where the rewards are. If you put the kindled rat back on the maze, it wanders around at random and actually doesn't find the rewards on many of the trials. So this suggests that hippocampal IEDs and their coupling with cortical spindles actually impairs specifically long-term memory. So importantly, we wanted to determine whether these findings are relevant to patients, because that's what we're all ultimately interested in. And to do this, we used intracranial EEG data from patients who are undergoing workup for epilepsy surgery. And as you all know, these patients are implanted with subdural grids, strips, and depths to try and localize their epileptic foci for potential resection. And so we found this same IED spindle coupling that we saw in our rodents in our patients, in our human subjects. And so what I'm showing here is an example, a raw trace. So here's an IED from one of our human subjects. And you can see, just like in the rats, we have this coupled spindle oscillation. And we can look at that in terms of our time frequency measures, and also, most importantly, in terms of our cross-correlation measure. So here again, time 0 is the time of the IED. You can see this nice significant peak of spindle coupling in our human subjects. And what we can do is we can map this across the surface of the brain. So here we take the amount of significant correlation from these cross-correlograms and we translate it into a color map. And then we plot the color across the surface of the brain. So for instance, in this patient, you can see that there's a high degree of IED spindle coupling that's located in this region here. And so in this way, we can map in our human subjects the cortical areas that are playing a role or are recruited by IED spindle coupling into the network. And so we wanted to gain some insight into the anatomic basis for these sorts of interactions. And so what we did was we examined the pairs of IED generating and spindle responsive brain regions. And so we designated a pair if IEDs in one brain region resulted in a significant cross-correlation with spindles in another brain region. And so what we found, very interestingly, was that IED spindle pairs often spanned more than one lobe. So this wasn't a local phenomenon. It was actually affecting large-scale interactions. And when we actually quantified this by measuring the distance Euclidean between the electrodes, we found that on average, the distance between the IED and its evoked spindle pair electrode was about 6 centimeters. And so this pathologic coordinated brain activity spans medium to long distances rather than remaining a local network phenomenon. So how does this relate to the actual seizure network? And so what we found here is that so here's an example of one of our patients showing a seizure. And so this is the red and the orange electrodes show the seizure onset. The seizure is subsequently recruited through the green electrodes, and then the blue electrodes are unaffected. And what we found that was a bit surprising to us is that the regions that expressed prominent spindle coupling with IEDs were actually in the blue, so outside of the ictal network. And so these sorts of IED spindle interactions are distinct from the ictal network and represent a way by which the epileptic activity is potentially reaching outside and affecting brain regions that are outside the epileptic network proper. So what happens to these sorts of interactions over time? So we're not able to answer this question very well in patients easily because in general, we don't have repeat intracranial EEGs in very many human subjects, but we can address it quite well in the Kindling model. And that's because the seizures increase in severity over time with the more stimulations you give. So initially the seizures are focal, but they progress to bilateral convulsions. And in parallel, if you look at IEDs in the structure that you're stimulating, the hippocampus, they become more and more and more over time. But what happens to brain regions that are outside of this epileptic zone? So when we looked at the MPFC, initially there were no IEDs in the MPFC proper, which makes sense because we're not stimulating there. But over time, with increasing epileptic activity in the hippocampus, we actually found that we started to get IEDs independently in the MPFC. So it suggests that there's actually a spreading or a recruitment of the MPFC as an independent interictal epileptic focus. And so to look at that, these are just some examples, and to show you early on during the Kindling, you can see that there's just in the hippocampus IEDs and nothing in the MPFC, and then a little bit later you start to have a tiny little IED that happens at the same time as the hippocampal IED, and pretty much all of this activity is strongly linked together. But late in the Kindling, what we found is that there were many, many times when the IEDs in the hippocampus occurred and there were separate IEDs in the MPFC. And if you look at the cross-correlogram, a lot of the activity still occurs together, but there's all of this activity outside here, which is independent MPFC interictal activity. So this suggests that we've actually recruited another brain region, perhaps perhaps similarly to what we saw in the previous talk. So how could this actually be related to LGS? And so I'm very glad that I'm speaking second, because we already heard a lot of evidence and information about how LGS is most likely a progressive disorder, and how you can have interictal patterns that come on earlier before you have refractory seizures, the relationship with focal discharges and focal lesions, and also the improvements that can happen after resective epilepsy surgery in patients with LGS who have focal lesions. And of course, we know that intellectual dysfunction can increase over time in LGS and actually affect a larger proportion of patients over time. And so what I think, or what I'm putting forward a little bit speculatively, is that a process similar to Kindling could actually play a role in the creation of large-scale network dysfunction in LGS. And so what could we do to manipulate these types of large-scale networks? So what we've been trying in our group is actually closed-loop electrical stimulation. And so this is a process where you detect an event of interest, and then you deliver a pulse of electrical stimulation back to the brain to try and modulate its function. And a major barrier for us so far is that IEDs are extremely brief events, and typically by the time you've detected them, there's not much you can do to modulate the network, because the IED has already affected monosynaptically connected structures. But from our work, we saw that IEDs couple with spindles, and this is a much more extended interaction. And some of the dysfunction in the network is being induced by this IED spindle coupling. This gives us enough time to actually have a network intervention to prevent the pathologic coupling. And so here's an example of our closed loop electrical stimulation that we that we devised in our lab. And so these are some of the electrodes that we implant in the rat's brain, and they're connected up to this little microprocessor that allows us to detect events and then deliver electrical stimulation. And so here's an example of our recording setup. And so when there's an IED in the hippocampus, such as in black right here, we actually have a microprocessor embedded that's going to give us this peak in signal, and when it crosses the threshold, this is a positive detection for an IED in the hippocampus. And so this allows us to have really low latency detection of relevant events on the order of approximately 15 milliseconds. So what we've been doing is working to understand how we can best interact with the network in a meaningful manner using this electrical stimulation. So this is an example of the unstimulated state. Here you have an IED in the hippocampus. This is a recording in the cortex where you see you have the coupled spindle oscillation. And if you plot their correlation, you see there's this high amount of pathologic connectivity between the hippocampus and cortex. Now, what we found after a lot of experimentation is that if you deliver a Gaussian wave of electrical stimulation when you detect a hippocampal IED, you can actually prevent a spindle from being expressed in the cortex. And so this is an example of a session where we've done this closed loop stimulation, and you can see when we plot all of our IED times, we have eliminated the increase or the pathologic coupling between the two structures. And so a clue to what might be going on here mechanistically actually comes from work with another group and actually extends the significance to LGS, because this group was working on a rodent model of spike wave discharges. And what they did is they detected the epileptic spikes and then delivered Gaussian stimulation immediately afterwards. And what they found is that actually decreased the duration of spike wave episodes in their rodents. And so the thinking here is that we're able to disrupt the pathologic synchronization by delivering this Gaussian stimulation such that repetitive activity, such as repetitive spike wave discharges or a spindle oscillation, is prevented from occurring. All right, so I wanted to share with you just a little bit of work, some results that we have. This is all preliminary data that is still actively ongoing in the group. But what we've done is we've taken some of our rats and we've actually put them on a Kindling and closed loop therapy to see what happens. And so what I'm showing you here are some examples. This is a rat that had Kindling only in the top row, and this is early in the Kindling when it only has focal seizures. You can see that there's some IEDs in the green channel, which is the hippocampus, but the cortex looks relatively preserved in terms of its signal, no pathologic activity. But late in the Kindling, you can see that there are multiple IEDs in the hippocampus. There are also independent IEDs in the cortex, and the network looks quite pathologic on this larger scale. So now this down here at the bottom, this is a rat that's had Kindling, exactly the same Kindling as this rat, but it's also had closed loop therapy every day in addition to its Kindling procedure. And so early on in the Kindling, you can see there are multiple intrarectal discharges in the hippocampus, none yet in the cortex. But after 2 weeks of closed loop therapy and ongoing Kindling, we actually have a dramatic reduction in the amount of pathology in the network, and the animal actually has not developed progression of the epileptic seizure semiology either. So we have a lot more work to do here, but I think it's a promising early result for what this type of intervention could do. So in conclusion, what I've tried to show you today is that intrarectal epileptic activity induces pathologic large-scale network interactions. This happens, we found this interaction in rodents, but it also happens in humans with epilepsy, and it contributes to memory dysfunction. And the progression of epilepsy in our models can result in creation of independent cortical foci, and this may be how epilepsy can spread and recruit more areas as it progresses, and that there is some promise for closed-loop neurostimulation to try and counteract these pathologic network interactions. And now, of course, this is all more speculative because, of course, none of this work specifically has been done in a model of LGS or patients with LGS. But the reason that I think that it may be quite relevant is that spike and slow wave patterns are very common in LGS, and so the mechanism of counteracting the coordinated network silence of the slow wave with the Gaussian stimulation that we've been using could potentially prevent the subsequent depolarization and expression of further epileptic spikes. And so in this manner, there could be an opportunity to disrupt the persistent intrarectal network pattern seen in LGS. And furthermore, it's possible that these sorts of approaches could prevent further cortical recruitment into the epileptic network. And so in this way, you could hope to protect cortices that are outside the initial epileptic network and increase the likelihood that targeted procedures such as a resection could have a good outcome. And there's also the potential to be able to restore physiologic sleep rhythms and coupling between physiologic oscillations, which is, of course, would be important for memory. And these all remain hypotheses, but I think that preliminary evidence is building regarding the mechanisms and potential efficacy. And I'm looking forward to collaborating in this sort of work in the future. And of course, I'd like to acknowledge all of our trainees and collaborators and funding agencies without which this work would be impossible. And if we have a few moments, I'm happy to take questions. Otherwise, happy to address them in the end. Thank you.
Thank you so much. That was wonderful. Yeah, we do have time for at least one question. If I'm not totally sure I'm going to be able to see if it's coming in and where it's coming from, but you can unmute yourselves, I believe.
If anyone has a question, they can raise their hand on the Zoom meeting. And I'm looking at the chat from the Google Doc, Brenda, so I'll see if anyone comes in there.
Okay.
Oh. So if anyone does not have a question, I'll ask one to Jennifer. With your type of white noise stimulation, can have you thought about pushing the system either to worsening those pathologic connections that are being generated in the animal by giving other stimuli, and how you can modulate it to kind of prove causality of this type of connection?
Yeah, that's a great question. And so in figuring out how to prevent the pathologic coupling, we came across many protocols that actually increase and worsen it first. And so we know the types of stimulation that we can deliver to increase the coupling. And I think that is a great set of experiments to show that, potentially we should be able to push the network in either direction, delay the progression of epilepsy or even accelerate it by increasing these types of abnormal interactions.
And then a follow-up, does the type of stimulation that you're providing to prevent that progression, what does it tell you about the underlying cellular electrophysiology or local circuit dynamics by having to give just kind of a white noise stimulation like that?
Yeah, so what we think is, or actually the rationale between the type of stimulation that we give is actually that we noticed that in the cortex, once and I didn't show this data, I didn't quite have time, but if you look at the neural spiking activity patterns of cortex, when they first perceive the intrarectal discharge from the hippocampus, there's a big increase in firing of the pyramidal cell neurons, and then both neuron populations, pyramidal cells and interneurons, decrease their firing, for a few hundred milliseconds, and then there's a rebound firing of both pyramidal cells and interneurons, and that's associated with the spindle oscillation. And so what we thought is that coordinated network silence is probably really important for priming this subsequent depolarization. And so we thought that potentially if we give a stimulation that causes some of the cells to start to fire during this period where they otherwise would have been silent, then some of them would be refractory and unable to participate in the subsequent rebound depolarization. And so in that way, you can actually interrupt the synchrony of the network, actually by causing cells to fire when they otherwise would have been silent. And the way that you do that is really important, because if you give a super strong pulse or a train of stimulation, you cause too many cells to fire all at once. And so you're actually, even triggering more coordinated activity. Whereas if you give this kind of graded, slow response, the cells are naturally going to fire based on their properties, but spread out over a longer period of time. And so therefore, it makes the network more refractory to any sort of coordinated subsequent activity.